close your eyes for a moment and imagine something. Imagine a child. A beautiful, healthy child, maybe 5 or 6 years old, running through a habitat on Mars. The red dust outside the windows, the thin carbon dioxide atmosphere pressing against the reinforced glass, and this child, born on another world, laughing and playing just like any child on Earth. Now imagine that same child, 15 years later, stepping off a spacecraft onto Earth for the very first time, and the moment their feet touch the ground, the moment full Earth gravity wraps around their body, their spine begins to compress in ways it was never designed to handle. Their knees buckle. Their vertebrae, thin and fragile like porcelain, crack under the weight of their own torso. Their heart, small and weak, struggles desperately to pump blood upward against a gravitational force it has never experienced. Within minutes they collapse. Not from illness, not from injury, but because their body was built by a different world, for a different world, and Earth has become as hostile to them as the vacuum of space. This is not science fiction. This is not speculation for dramatic effect. This is the direct, logical, scientifically supported consequence of everything we know about human developmental biology, about how gravity shapes every single cell in your body from the moment of conception, and about what happens when you remove 62% of that shaping force during the most critical years of human growth. Today, we are going to take a journey through one of the most profound and least discussed problems facing humanity's future in space. We are going to go deep, system by system, through the human body, and I am going to show you exactly what Mars gravity, at just 38% of Earth's, would do to a child growing up on that planet. We will look at bones, muscles, the heart, the brain, the eyes, the spine, the lungs, the immune system, the vestibular system, and more. We will examine real research from real scientists at institutions like the University of Calgary, NASA, the European Space Agency, Johns Hopkins University, and dozens of other research groups around the world. We will look at data from the International Space Station, from ground-based analog studies, from animal experiments, and from clinical research on Earth that gives us terrifying clues about what reduced gravity does to growing bodies. And here is what makes this truly fascinating. We are not talking about science fiction scenarios set hundreds of years in the future. We are talking about something that could happen within our lifetimes. Elon Musk talks about sending humans to Mars within this decade. SpaceX is building the Starship right now. NASA has an Artemis program designed to return humans to the Moon as a stepping stone to Mars. China has announced Mars mission timelines. The conversation about colonizing Mars is not theoretical anymore, it is operational. And yet almost nobody is talking about the single most important question: what happens to the children? Because here is the thing about colonization. It is not a colony if people just visit. It is not a colony if adults go there, work for a few years and come back. A real colony means families. It means pregnancy. It means birth. It means children growing up, developing, going through puberty, reaching adulthood. And every single one of those processes is shaped, guided, and fundamentally determined by gravity. Remove the gravity, or reduce it by 62%, and you do not just make people lighter. You fundamentally alter the developmental blueprint that 4 billion years of evolution has written into every cell of the human body. Let me ask you some questions that I want you to hold in your mind throughout this entire discussion. Why do your bones have the exact density they have? Why is your heart the exact size and strength it is? Why does your spine have exactly 4 curves? Why are your muscles exactly as strong as they are? The answer to every single one of these questions is the same. Gravity, Earth's gravity specifically, 9.8 1 meters per second squared. 1G. The constant, invisible, relentless force that has shaped human biology since the first vertebrate crawled out of the ocean onto land 375 million years ago. Now, before we dive deep into the science, let me share with you a research story that will set the foundation for everything we are about to discuss. Because this is not my opinion. This is not speculation. This is peer-reviewed, published, replicated science. In 2015, a scientist named Dr. Stephen Boyd, the director of the McCaig Institute for Bone and Joint Health at the University of Calgary in Canada, launched one of the most ambitious bone studies ever conducted on astronauts. He called it the T-Bone Study. Over the next 7 years, Boyd and his team, led by Dr. Lee Gabel, an assistant professor in kinesiology, would follow 17 astronauts through an incredible journey. They would scan these astronauts' bones before they left for the International Space Station, immediately upon their return to Earth, and then again at 6 months and 12 months after landing. But this was not just any bone scan. Boyd's team used a technology called second-generation high-resolution peripheral quantitative computed tomography. This machine can image bone structure at a resolution of 61 micrometers, that is 61 millionths of a meter. It can see individual trabeculae, the tiny rod-like structures inside your bones that give them their strength. It can measure cortical thickness, the dense outer shell. It can calculate bone failure load, which is literally how much force a bone can take before it breaks. The team traveled repeatedly to NASA's Johnson Space Center in Houston, Texas. They scanned the wrists and ankles of each astronaut at 4 time points. And what they found, published in June 2022 in Scientific Reports, a journal by Nature, was genuinely shocking. Here is what the data showed. After spending 4 to 7 months on the International Space Station, astronauts experienced significant bone loss in their weight-bearing bones. That was expected. What was not expected was what happened next. After 12 months back on Earth, with full gravity, with access to exercise, with proper nutrition, with medical support, 9 out of 17 astronauts showed incomplete bone recovery. Their bones did not come back to what they were before the flight. For the 8 astronauts who had spent more than 6 months in space, the bone loss was equivalent to approximately a decade of normal aging on Earth. And much of it appeared to be permanent. Let me say that again, because I need you to understand the magnitude. 6 months in reduced gravity caused bone damage equivalent to 10 years of aging, and it did not fully recover even after a year back on Earth. Dr. Gabel said, and I quote, "We found that weight-bearing bones only partially recovered in most astronauts 1 year after spaceflight." This suggests the permanent bone loss due to spaceflight is about the same as a decade worth of age-related bone loss on Earth. But here is the detail that should truly terrify you. The researchers found that the bone loss was not just about density, it was about architecture. Inside your bones, there is a complex network of tiny rod-like structures called trabeculae. Think of them like the internal scaffolding of a building. These rods are connected to each other, forming a mesh that gives your bones their ability to resist force from multiple directions. When astronauts lost bone in space, some of these trabeculae thinned to the point where they disconnected from the network. And here is the critical point. Once a trabecular rod disconnects, it cannot reconnect. The architecture is permanently altered. Even if new bone mineral is deposited during recovery, it deposits on the remaining trabeculae, making them thicker. But it cannot rebuild the connections that were lost. The geometry of the bone is permanently changed. Now I need you to pause and think about what this means. These were adult astronauts. Adults, their bones were fully formed, fully mineralized, fully developed over decades of living in Earth's gravity. They had reached peak bone mass. They had strong, healthy, mature skeletons. And even so, just a few months of reduced gravity caused permanent, irreversible damage to their bone architecture. Now imagine, just imagine, what would happen to a child whose bones are not fully formed? A child whose skeleton is actively growing, actively developing, actively building its architecture for the first time? A child whose growth plates are still open, whose bones are still deciding how dense to be, how thick to be, how strong to be. And instead of receiving the full signal of Earth's gravity telling those bones to build strong, they receive only 38% of that signal. This is the question we are going to answer today. And the answer is profound, disturbing, and absolutely fascinating from a scientific perspective. If you find this kind of deep exploration fascinating, if you want to understand the real science behind space colonization that nobody else is talking about, then subscribe to this channel and hit the notification bell. Drop a comment below and tell me, knowing what you are about to learn. Would you allow your child to be born on Mars? Let us continue. To understand what Mars gravity would do to a developing child, we first need to understand something fundamental about how your body works. And this might change the way you think about gravity forever. You probably think of gravity as just a force that keeps you on the ground, something that prevents you from floating away. Something passive, simple, boring even. But that is profoundly wrong. Gravity is not just a force, it is a signal. It is a developmental instruction. It is, in the most literal biological sense, a sculptor. And your body is its sculpture. Every single second of every single day, from the moment you were conceived to this very moment right now, gravity is talking to your cells. It is sending mechanical signals through your bones, your muscles, your connective tissues, your organs. And those cells are listening. They are responding. They are building and rebuilding themselves based on what gravity tells them. This is not a metaphor. This is measurable, quantifiable, proven biology. Let me explain how it works. In 1892, a German anatomist and surgeon named Julius Wolff published a paper that would become one of the most important principles in orthopedic medicine. It is called Wolff's Law, and it states something elegantly simple. Bone in a healthy person will adapt to the loads under which it is placed. If loading on a particular bone increases, the bone will remodel itself over time to become stronger to resist that sort of loading. If the loading decreases, the bone will become weaker because the stimulus for remodeling is not present. This is not a theoretical principle. This is an observable, measurable fact. Tennis players, for example, have been shown to have 20 to 40% greater bone density in their dominant arm compared to their non-dominant arm. Same person, same genetics, same nutrition, same hormones, but different mechanical loading on each arm. And the bone responds by being denser and stronger where it is loaded more. Your body does not waste resources. Building bone is metabolically expensive. It requires calcium, phosphorus, collagen, energy. Your body will only build bone where it is needed, to the degree it is needed, as determined by mechanical loading. And the single biggest source of mechanical loading on your skeleton is gravity. The constant downward pull of Earth's 9.81 meters per second squared acting on your body mass, transmitted through your skeleton every time you stand, walk, run, jump, or simply sit upright. Now, in 1987, an American orthopedic researcher named Harold Frost expanded on Wolff's work with what he called the mechanostat theory. Frost proposed that bones have a set point for mechanical strain, similar to how a thermostat has a set point for temperature. If the strain on a bone falls below a certain threshold, the bone will lose mass. If the strain exceeds a certain threshold, the bone will gain mass. The bone is constantly adjusting itself to maintain the minimum adequate strength for the forces it experiences. This is beautiful and elegant in its logic. Why would your body maintain bones stronger than they need to be? That would waste energy and resources. And why would it maintain bones weaker than they need to be? That would lead to fractures and death. So the body uses gravity and mechanical loading as the input signal to determine exactly how strong each bone should be. And here is the key insight for our discussion. If you take a developing child and place them in an environment where gravity is only 38% of what their genetic blueprint expects, their mechanostat will calibrate to that reduced loading. Their bones will reach equilibrium at Mars gravity levels. They will build bones that are perfectly adequate for Mars. But those same bones will be catastrophically inadequate for Earth. Let me go even deeper into the cellular mechanism, because this is where it gets truly amazing. Inside your bones, embedded within the mineralized matrix, there are cells called osteocytes. These are former bone-building cells that became trapped within the bone they created, and they now serve as the bone's sensory system. Each osteocyte has long branching extensions called dendritic processes that reach out through tiny channels in the bone called canaliculi. These processes connect osteocytes to each other, forming a vast sensory network throughout the entire skeleton. When gravity loads your bones, the bone matrix deforms ever so slightly. This deformation, this micro-strain, is detected by the osteocyte network. The bending of the bone causes fluid to flow through the canaliculi, and the osteocytes detect this fluid flow through their dendritic processes. This is called mechanotransduction. The conversion of a mechanical signal, gravity acting on your bones, into a biological signal that tells cells what to do. When osteocytes detect adequate strain, they send chemical signals that maintain the balance between bone building and bone breaking. They regulate a signaling pathway called the Wnt pathway, which promotes bone formation. They also regulate a molecule called rankle and another called OPG, which together control bone resorption. The process by which specialized cells called osteoclasts break down bone. In normal gravity, adequate strain causes osteocytes to activate Wnt signaling, promoting bone formation, and to produce OPG, which inhibits rankle and thus reduces bone breakdown. The result: strong, well-maintained bones. In reduced gravity, inadequate strain causes osteocytes to down-regulate Wnt signaling, reducing bone formation, and to produce more rankle while producing less OPG, increasing bone breakdown. The result: bones actively dissolve themselves because the signal says they are unnecessarily strong. This is not a malfunction. This is the system working exactly as designed. The bones are responding correctly to the information they are receiving. The problem is that the information, coming from a low-gravity environment, is telling the bones they do not need to be strong. And in a developing child, this means the bones never build their full potential in the first place. Let me give you the numbers. On Earth, a 30-kilogram child experiences a gravitational force of approximately 294 newtons, pulling downward through their skeleton. On Mars, that same child would experience only about 112 newtons. That is 182 newtons less force. 62% less mechanical loading signal reaching every osteocyte in their body. 24 hours a day, 7 days a week, throughout their entire developmental period from birth through adolescence. And here is something we need to talk about that makes this even more concerning. In 2023, a research team led by Jaap Swanenberg at the University of Zurich published a study in the journal Frontiers in Physiology examining what happens to the human body in simulated Mars gravity versus microgravity. They used a specialized apparatus to simulate zero gravity lunar gravity, and Mars gravity, measuring the response of spinal motor control. Their finding was remarkable and troubling. Mars gravity, at 0.38 g, induced similar changes in spinal stiffness and muscle deactivation as full microgravity. Let me repeat that. Mars gravity was not significantly better than zero gravity when it came to spinal motor control changes. The researchers found that even at Mars-level gravity, the transversus abdominis muscle, one of the key deep core stabilizers, showed decreased activity similar to what happens in total weightlessness. This suggests something deeply important. Mars gravity might not be enough. It might not provide a sufficient mechanical signal to maintain or develop normal human musculoskeletal function. When we talk about Mars gravity being 38% of Earth's, we might imagine that it provides 38% of the developmental signal. But biology is not linear. There may be threshold effects. Below a certain gravity level, the developmental signals might not just be reduced proportionally, they might effectively shut off. And Mars may be below that threshold for many critical developmental processes. Now let us go systematically through the human body, system by system, and examine what we know about how Mars gravity would affect a developing child. We are going to start with the skeletal system, because it is the most studied and provides the clearest picture. The development of the human skeleton is one of the most complex and gravity-dependent processes in biology. It begins in the 8th week of fetal development and continues until the mid-20s. There are two fundamentally different ways that bones form in the human body. The first is called intramembranous ossification, which is how flat bones like the skull and some facial bones form directly from mesenchymal tissue. The second is called endochondral ossification, which is how long bones like the femur, tibia, humerus, and others form through a cartilage template that is gradually replaced by bone. For our discussion, endochondral ossification is particularly important because it involves structures called growth plates or epiphyseal plates. These are zones of cartilage located near the ends of long bones where bone lengthening occurs. Throughout childhood and adolescence, cells within the growth plates divide, mature, and are gradually replaced by bone. This is how you grow taller. This is how your long bones reach their adult length. And here is the critical point. The behavior of growth plates is profoundly influenced by mechanical loading. There is a principle in orthopedics called the Hueter-Volkmann principle, which states that increased compression on a growth plate slows its growth rate, while decreased compression allows faster growth. This might seem to suggest that Mars children would grow taller, which may indeed be true. But there is much more to the story. Mechanical loading does not just influence how fast bones grow, it influences how they grow. The architecture, the density, the cross-sectional geometry, the cortical thickness, the trabecular network, the mineral content per unit volume, all of these are regulated by the forces acting on the bone during development. A bone that grows quickly under minimal loading does not grow the same way as a bone that grows under proper loading. It may be longer, but it will be thinner. It may be wider in diameter, but with a thinner cortex. It may have a larger marrow cavity, but less actual bone material. Think of it like this. You can grow a tree very quickly by giving it lots of water and nutrients in a greenhouse with no wind. But that tree will have a thin, weak trunk because it never needed to resist the force of wind. The moment you take it outside into a storm, it snaps. The tree grew, but it did not grow strong. Let me share some research that illustrates this principle in a real-world context. Studies on children with severe cerebral palsy provide one of the closest analogues we have to understanding what reduced mechanical loading does to developing bones. These children live in Earth's gravity, but because of their neurological condition, many cannot walk, cannot stand, and cannot properly load their bones through physical activity. A 2025 study by Al-Jishi and colleagues, published in a major orthopedic journal, found that children with moderate to severe cerebral palsy who had limited ambulation exhibited significantly lower bone mineral density compared to children who could walk. Another 2025 study by Zimmerman and colleagues, published in the Journal of Bone and Mineral Research, found that children with cerebral palsy had smaller bone cross-sections than typically developing children, and that walking ability was directly correlated with bone mass. These children experience full Earth gravity. They just cannot load their bones properly through movement. Now imagine a child who lives in only 38% gravity. Even if they run, jump, and play as actively as any child on Earth, the maximum force their activities can generate is only 38% of what an Earth child generates. Every jump produces only 38% of the impact. Every step transmits only 38% of the ground reaction force. The bones never receive the signal to build to Earth standard strength. Now let us talk about peak bone mass, because this is perhaps the most critical concept for understanding the long-term consequences. Throughout childhood and adolescence, your body is building what scientists call your bone bank. You are depositing bone mineral, building trabecular architecture, thickening cortical shells, and strengthening your skeleton. This process reaches its maximum, called peak bone mass, between the ages of 25 and 30. After that, bone mass gradually declines for the rest of your life. How strong your bones are at their peak determines your lifetime fracture risk. It determines whether you will develop osteoporosis in old age. It determines how much bone you can afford to lose before you become fragile. 90% of peak bone mass is achieved by age 18. The entire trajectory of your skeletal health for the rest of your life is largely determined by what happens during childhood and adolescence. Now, scientific models based on what we know about bone mechanobiology suggest that a child developing in Mars gravity would achieve only 40 to 60% of normal peak bone mass. Think about what that means. A Mars child would enter adulthood with bones that are already at the level of a 70 or 80-year-old Earth human. They would begin their adult life in what we would classify on Earth as the osteoporotic range. And then, normal aging would reduce their bone mass even further from that already diminished baseline. By their 30s or 40s, they might have the skeletal fragility of a 90-year-old. And here is the part that makes this truly irreversible. Unlike adult astronauts who lose existing bone and can potentially regain some of it, a Mars child would never build that bone in the first place. You cannot regain what was never formed. Once the growth plates close in late adolescence, once the window of peak bone mass development closes, you cannot go back and rebuild the architecture. Those trabecular rods that never formed cannot be created later. The cortical thickness that was never laid down cannot be added after the fact. The bone's fundamental structural blueprint is set during development, and if that development occurred in reduced gravity, the blueprint is permanently inadequate for Earth. A follow-up study published in 2025 by Matheson and colleagues, examining astronauts' bones 4 years after spaceflight, confirmed that even 4 years later, bone architecture had not fully recovered. And these were adults with mature skeletons who returned to full gravity. A child who never experienced full gravity would have an even more permanent deficit. Now let us move to the spine, because the spine is perhaps the most dramatic example of a structure that is literally sculpted by gravity. The human spine is not straight. It has 4 distinct curves. The cervical curve in the neck, which curves forward. The thoracic curve in the upper back, which curves backward. The lumbar curve in the lower back, which curves forward. And the sacral curve at the base, which curves backward. These 4 curves work together like a spring, absorbing shock, distributing load, and maintaining balance over the center of gravity. Here is what most people do not know. These curves are not pre-programmed at birth. A newborn baby has essentially a single C-shaped curve to its spine. The 4 adult curves develop after birth, in direct response to gravity and movement. The cervical curve develops at around 3 to 4 months of age, when the baby first begins to hold its head up against gravity. The lumbar curve develops at around 8 to 12 months, when the baby begins to sit upright and eventually stand. These are not genetically timed events. They are responses to gravitational loading. The spine curves because gravity is pulling on it, and the baby's muscles are fighting against that pull. On Mars, with only 38% gravity, these developmental forces would be dramatically reduced. The cervical curve might develop, but less pronounced. The lumbar curve might develop later and with less curvature. The overall spring-like architecture might be significantly different. A child whose spine develops in Mars gravity would have a fundamentally different spinal geometry than any human who has ever lived on Earth. But the problem goes deeper than curve development. Let me tell you about what happens between the vertebrae. Between each pair of vertebral bones, there is a structure called an intervertebral disc. These discs have 2 components, a tough outer ring called the annulus fibrosus and a gel-like center called the nucleus pulposus. On Earth, gravity compresses these discs throughout the day. When you lie down at night, the discs reabsorb fluid and swell slightly, which is why you are slightly taller in the morning than at night. In space, without gravity to compress the discs, they absorb significantly more fluid and swell substantially. This is why astronauts grow up to 5 centimeters taller in space. Their spines elongate because the discs are no longer being compressed. When they return to Earth, gravity recompresses the discs and they return to their normal height over a few days. Now think about a child developing on Mars. Their intervertebral discs would develop under only 38% of normal compressive force. The annulus fibrosus, that tough outer ring, develops its strength in response to the forces it must resist. Less force means less strength development. The nucleus pulposus, the gel center, would maintain a more hydrated, swollen state as its baseline. The entire structure of each disc would be designed for Mars-level compression. This has several consequences. First, the child would likely be taller than an equivalent Earth child, not because of more growth, but because of less spinal compression. Second, the discs would be structurally weaker, with a thinner, less organized annulus fibrosus. Third, if that person ever experienced Earth gravity, the sudden full compression on these inadequately developed discs could cause immediate herniation, where the nucleus pulposus bursts through the weakened annulus and presses on spinal nerves. It gets worse. Astronauts already have approximately 4 times the rate of disc herniation compared to the general population after returning from space. 4 times. And these are people whose discs were fully formed in Earth gravity before going to space. A child whose discs developed in Mars gravity would have even weaker disc structure and even higher herniation risk if gravity increased. And then there is the matter of spinal muscles. The human spine does not hold itself upright by bone structure alone. It requires a complex system of muscles that constantly contract to maintain posture against gravity. The deepest of these are the multifidus muscles and the transversus abdominis. These muscles develop their strength and endurance through the constant work of maintaining posture against gravity throughout childhood. Remember that 2023 study by Swanenberg? They found that even Mars gravity caused similar decreases in transversus abdominis activity as microgravity. If these critical spinal stabilizing muscles are not being adequately stimulated during development, they would never develop proper strength and coordination. A child growing up on Mars might have chronically weak, deep spinal muscles, potentially leading to spinal instability, abnormal movement patterns, and increased risk of injury even in Mars gravity as they age. Let me take you through the muscular system more broadly now, because what happens to muscles in reduced gravity is almost as concerning as what happens to bones, and the two systems are intimately connected. Your muscles are not a fixed quantity determined by your genes. They are living, adaptive tissues that constantly adjust their size, strength, and fiber composition based on the demands placed on them. And the single greatest demand placed on your muscles, outside of deliberate exercise, is the constant work of opposing gravity. Think about it. Every moment you are standing, your calf muscles, your quadriceps, your gluteal muscles, your spinal erectors, your core stabilizers are all working to keep you from collapsing to the ground. Even when you are sitting, your trunk muscles are working to hold your spine upright. These muscles that work against gravity are called anti-gravity muscles or postural muscles, and they represent a huge proportion of your total muscle mass. On the International Space Station, astronauts lose approximately 20% of their muscle mass in just 5 months, despite exercising for 2 hours every day using specialized equipment. 2 hours of daily exercise is not enough to counteract the loss of 24-hour gravitational loading. In 2024, Mergia and colleagues published a study in Nature examining the skeletal muscle proteome of astronauts before and after spaceflight. They found fundamental changes in muscle protein composition, not just atrophy, but actual alterations in the molecular makeup of the muscle tissue. Different proteins were being expressed. The ratio of muscle fiber types was changing. The muscle was not just getting smaller, it was becoming a different kind of tissue. Human muscles contain 2 main types of fibers. Type 1 fibers, also called slow twitch fibers, are designed for endurance and sustained contractions. These are the fibers that dominate in your postural muscles. Type 2 fibers, or fast twitch fibers, are designed for power and quick movements. In microgravity, and likely in Mars gravity, it is the type 1 fibers that atrophy preferentially. The sustained postural work disappears, so the body reduces the fibers designed for that work. Now here is what matters for children. Muscle fiber type is not fully determined at birth. During early childhood, muscle fibers are still differentiating, still committing to becoming type 1 or type 2. This process is influenced by the demands placed on the muscle during development. A child developing in Mars gravity would have reduced demand for type 1 postural fibers. Their muscles would likely develop with a different fiber type ratio, a lower proportion of the slow twitch endurance fibers that maintain posture against gravity. This is not a problem that can be fixed with exercise. Once muscle fiber type composition is established during development, it is very difficult to change in adulthood. An adult can make existing fibers larger through exercise, but changing the fundamental ratio of fiber types is extremely limited. A Mars child whose muscles developed with the wrong fiber ratio would carry that limitation for life. But here is where it gets truly fascinating from a systems biology perspective. Muscles and bones do not develop independently. They develop as an integrated functional unit. Muscles pull on bones through tendons. This pulling creates additional mechanical loading on the bones, which stimulates bone growth. Stronger muscles create stronger forces on bones, which signal for stronger bones to be built. This is a positive feedback loop that drives the development of the entire musculoskeletal system. In Mars gravity, this feedback loop operates at a much lower level. Muscles develop to only 38% of Earth's standard strength, because that is all gravity demands. Those weaker muscles generate only 38% of the force on bones. The bones receive even less mechanical stimulation and develop even weaker. The weaker bones provide a weaker scaffold for muscle attachment, further limiting muscle force production. The entire system settles into a low development equilibrium that is perfectly functional for Mars but catastrophically inadequate for Earth. A child running and jumping on Mars at maximum physical exertion generates forces on their bones and muscles that are roughly equivalent to a very sedentary, bedridden person on Earth. Their most active moments produce loading signals that would be classified as dangerously insufficient for skeletal maintenance in an Earth clinical setting. Their body is living in what any Earth physician would recognize as a disuse state, even when they are playing as hard as they can. Let me tell you something about proprioception now, because this is another system that would be fundamentally altered, and it affects everything from balance to coordination to how a person moves through the world. Proprioception is your body's sense of where it is in space. Not outer space, but physical space. It is how you know where your hand is without looking at it. It is how you can walk without watching your feet. It is how you maintain balance without consciously thinking about it. This sense comes from specialized receptors in your muscles, tendons, and joints. The two key proprioceptive organs are muscle spindles and Golgi tendon organs. Muscle spindles are tiny sensory structures wrapped around individual muscle fibers that detect stretch and length changes in the muscle. Golgi tendon organs are located at the junction between muscles and tendons and detect the force being generated by muscle contraction. Together, these sensors give your brain a constant stream of information about the position and movement of every part of your body. Here is the critical point. These sensory systems are calibrated during development. They learn what normal feels like based on the gravitational environment they develop in. The sensitivity settings, the reflex thresholds, the neural integration patterns. All of these are established during childhood based on the constant feedback from living in a particular gravity. A child developing on Mars would have proprioceptive systems calibrated to 0.38 G. Their muscle spindles would be tuned to detect the range of stretches that occur during Mars-level movement. Their Golgi tendon organs would be calibrated to the range of forces produced against Mars gravity. Their cerebellum and motor cortex would wire themselves based on the sensory input from a Mars environment. If that person ever came to Earth, their entire proprioceptive system would be overwhelmed. Forces they had never experienced would be flooding through sensory organs that cannot process them properly. Their reflexes, tuned for Mars, would be completely inappropriate for Earth. Balance would be nearly impossible because the vestibular and proprioceptive integration was never calibrated for this level of gravitational input. Let us now talk about what Mars gravity would do to the cardiovascular system, because this is where the consequences become potentially lethal. Your heart is a muscle. Let me say that again because it is easy to forget. Your heart is a muscle. And like all muscles, it develops its size, strength, and endurance based on the demands placed upon it. The primary demand on your heart is pumping blood throughout your body against gravity. When you are standing upright on Earth, your heart must generate enough pressure to push blood approximately 1.5 meters straight up from your heart to your brain against the full force of gravity. This is not easy. It requires a thick, strong left ventricle capable of generating significant pressure. Your heart has developed over your lifetime to be exactly strong enough for this task. Consider the hydrostatic pressure column in your body. When standing, there is approximately 100 millimeters of mercury pressure difference between your head and your feet, purely due to gravity. Your heart must overcome this gradient to keep your brain perfused. Your blood vessels must be strong enough to withstand this pressure in your lower body. Your venous system must have one-way valves to prevent blood from pooling in your legs. Every component of your cardiovascular system is designed around this gravitational challenge. On Mars, the hydrostatic pressure column would be only 38% as large. Approximately 38 millimeters of mercury difference between head and feet instead of 100. The heart would need to generate much less pressure to perfuse the brain. The blood vessels would experience much less pressure in the lower body. The venous valves would need to resist much less gravitational pooling. For an adult who developed on Earth and then goes to Mars, this means their heart is overpowered for the environment. It was built for a harder job and now has an easier one. This leads to cardiac atrophy in astronauts. The heart shrinks because it is not being challenged. Studies have shown that in microgravity, the heart becomes more spherical and loses mass. It literally remodels itself to match the reduced demand. But for a child whose heart develops from the start in Mars gravity, the situation is fundamentally different. Their heart would never build to Earth specifications. It would develop to be exactly adequate for Mars. Smaller, thinner-walled, weaker, but perfectly functional in that environment. A 2024 study from Johns Hopkins University examining heart muscle cells in low-gravity conditions found that reduced gravity weakens heart muscle cells and disrupts their normal electrical rhythm. The cells became weaker and showed abnormal contractile patterns. Now, these were isolated cells in a laboratory setting, not a developing heart in a child. But the principle is clear. Cardiac tissue that develops under reduced gravitational demand develops differently. The heart develops rapidly during fetal life and the first years after birth. Cardiac muscle cells, called cardiomyocytes, undergo the majority of their multiplication during fetal development and the first year of life. After that, the heart grows primarily by each existing cell getting larger, not by adding new cells. The demands placed on the heart during this critical window of cell multiplication help determine the total number of cardiomyocytes and thus the heart's lifetime capacity. A fetus developing on Mars would experience reduced cardiovascular demands from the very beginning. The mother's circulation would already be adapted to Mars gravity. The fetal heart would develop in the context of reduced placental blood flow demands. After birth, the infant heart would face only 38% of the circulatory challenge it would face on Earth. The number of cardiomyocytes might be lower. The thickness of the ventricular walls would be less. The maximum cardiac output would be permanently limited. What would happen if this person with their Mars-developed heart stood up on Earth? The math is straightforward and terrifying. Their heart was designed to push blood 1.5 meters upward against 0.38 g. Now it must push blood the same distance against 1 g. The demand has increased by 2.6 times. But their heart was never built for this demand. It cannot generate the necessary pressure. The immediate consequence would be orthostatic hypertension, insufficient blood pressure to perfuse the brain when standing. The brain, starved of blood, would shut down. Unconsciousness within seconds. If somehow kept upright, if somehow supported, the heart would be working far beyond its design capacity. This is not like an athlete pushing to exhaustion. This is like asking a motor designed for a bicycle to power a truck. The motor does not just tire, it fails. And the blood vessels would be equally inadequate. Arterial walls in the lower body develop their thickness based on the pressure they must withstand. Less pressure means thinner walls. Earth-level hydrostatic pressure in vessels designed for Mars-level pressure could cause vascular damage or rupture. Veins in the legs, with underdeveloped valves, would allow massive gravitational pooling of blood. The cardiovascular system would fail at multiple levels simultaneously. This is not speculation. This is direct extrapolation from known physiology. We know that adult astronauts who spend just 6 months in space cannot stand up without fainting when they return to Earth, and their systems were built for Earth. A person whose cardiovascular system was never built for Earth would have a far more severe and potentially permanent inability to function in full gravity. Now let me take you to the brain and the eyes. Because what we have discovered about intracranial pressure in space is perhaps the most unexpected finding in space medicine history. In 2005, NASA flight surgeons noticed something disturbing. Several astronauts returning from the International Space Station were reporting blurred vision. Their eyes were being examined routinely before and after flights, and the doctors noticed something. The back of some astronauts' eyes was swollen. The optic disc, where the optic nerve meets the retina, was bulging forward. The back of the eyeball itself was being flattened, pushed inward, and the astronauts' vision had shifted, becoming more farsighted. What was causing this? After years of investigation, the answer became clear. Increased intracranial pressure. The cephalid fluid shift. In microgravity, without gravity to pull fluid down to the lower body, approximately 2 liters of fluid that would normally be in the legs migrates upward into the torso and head. This increases the pressure inside the skull, and that increased pressure pushes on the back of the eyeballs and on the optic nerves. NASA named this condition Spaceflight-Associated Neuro-Ocular Syndrome, or SANS. It is currently classified as NASA's #1 health risk for long-duration spaceflight. It affects approximately 40% of astronauts on long missions. A comprehensive review published in 2018 in the journal Eye and a 2026 update in Nature's Eye journal detail the clinical manifestations: optic disc edema, globe flattening, choroidal folds, and refractive changes. Part 2 now. What does this mean for children on Mars? Mars has less gravity than Earth, which means less downward pull on fluids. The cephalid fluid shift on Mars would be less extreme than in microgravity, but more extreme than on Earth. There would be chronically elevated intracranial pressure compared to what human biology evolved for. Not as severe as the IS, but persistently higher than normal. For an adult, this causes damage over months and years. For a developing child, the consequences could be far more profound. The human eye is still developing during childhood. A process called emmetropization, where the eye adjusts its shape to achieve proper focus, continues until approximately age 6 to 8. The shape of the eyeball is partially determined by the pressure inside and outside it. Chronically elevated intracranial pressure during this developmental window could permanently alter the shape of the eye, leading to lifelong refractive errors that no glasses can fully correct because the eye's fundamental geometry is wrong. The optic nerve is another concern. During childhood, the optic nerve is still developing its myelin sheath, the insulating layer that allows rapid signal transmission. Chronic pressure on the developing optic nerve could impair this myelination process, potentially leading to reduced visual acuity, impaired color vision, or even partial visual field loss. And then there is the brain itself. The human brain develops within a specific intracranial pressure range that has been maintained by evolution for millions of years. During early childhood, the brain is undergoing massive development. Neurons are forming connections at a rate of millions per second. Neural migration, where newly formed neurons travel to their correct location in the brain, is occurring. Myelination of neural pathways is progressing. The brain is literally wiring itself. This wiring process occurs within the physical constraints of the skull, guided by chemical signals, genetic instructions, and the physical environment, including the pressure of the cerebrospinal fluid surrounding the brain. If that pressure is chronically elevated, even slightly, during the most critical windows of brain development, the consequences could be subtle but significant. Altered neural migration patterns, different ventricle sizes, changes in cerebrospinal fluid circulation. We genuinely do not know the full implications because we have never had a developing human brain in reduced gravity. But there are clues from medicine on Earth. In children with untreated hydrocephalus, where intracranial pressure is chronically elevated, we see white matter abnormalities, cognitive changes, and neurological impairment. Now, Mars gravity would not cause anything as severe as hydrocephalus. The pressure elevation would be much more subtle, but it would be present every moment of every day for the entire developmental period. Even a small chronic deviation from the evolutionary norm, sustained over years, could produce meaningful changes in brain structure and function. There is also the matter of cranial development. In infants, the skull is not a solid structure. It consists of separate bone plates connected by fibrous tissues called sutures, with gaps called fontanelles. This arrangement allows the skull to deform during birth and to expand as the brain grows. The timing of suture closure, the shape of the final skull, is determined by the interplay between brain growth pressure from within and gravitational forces from without. In reduced gravity, with altered intracranial pressure dynamics, the pattern of skull development could differ. The head might develop slightly larger or a different shape, or with altered proportions between the cranial vault and the facial bones. These are subtle effects, but over a full developmental period, they could produce noticeable differences in the appearance of Mars-born humans. Now let us talk about the vestibular system, because this is a system that is fundamentally, directly dependent on gravity for its very function. The vestibular system is located in your inner ear. It consists of two main components: the semicircular canals, which detect rotation, and the otolith organs, which detect linear acceleration and gravity. The otolith organs, the utricle and saccule, contain tiny calcium carbonate crystals called otoconia that sit on top of sensory hair cells. When your head tilts relative to gravity, these crystals shift, bending the hair cells and sending a signal to your brain about your orientation relative to the gravitational vertical. This system is structurally complete at birth, but its functional connections to the brain, the neural processing that converts vestibular signals into balance, posture, and spatial orientation, continues to develop throughout childhood and into adolescence. The system calibrates itself to the gravitational environment during this developmental period. In 2008, April Ronker and colleagues at NASA published a landmark study in which pregnant rats were flown on the space shuttle during the period when their fetuses' vestibular systems were developing. The offspring showed significant differences in vestibular function compared to ground control animals. Their vestibular systems had developed differently because they experienced microgravity during the critical window of vestibular development. A child developing on Mars would have a vestibular system calibrated to 0.38 G. Their otolith organs would function perfectly well for detecting tilt and acceleration on Mars. But on Earth, the same otolith organs would receive signals 2.6 times stronger than anything they were calibrated for. The brain's vestibular processing centers, wired during development for Mars-level input, would be overwhelmed and confused by Earth-level gravitational signals. The result would be severe vestibular dysfunction on Earth, constant vertigo, inability to maintain balance, spatial disorientation, nausea, and impaired eye movement control. The vestibulo-ocular reflex, which stabilizes your vision during head movements, would be improperly calibrated. A Mars person on Earth might not be able to read text while walking because their eyes cannot properly compensate for head motion against Earth-level gravitational bobbing. And here is what makes this particularly concerning. Unlike bones or muscles, which might theoretically be strengthened with extreme exercise programs, the vestibular system does not have a simple countermeasure. You cannot exercise your otolith organs into greater strength. Vestibular adaptation in adults is possible but limited, and the fundamental calibration set during development may not be fully reprogrammable in adulthood. Now I want to discuss the respiratory system, because here too, gravity plays a role that most people never consider. Your lungs are complex, delicate structures that are profoundly affected by gravity. On Earth, gravity creates uneven distribution of both air and blood within the lungs. The bases of the lungs receive more blood flow than the tops because gravity pulls blood downward. The distribution of ventilation is also gravity dependent. This creates what pulmonologists call ventilation perfusion matching, where the areas receiving the most blood also receive the most air. This matching is critical for efficient gas exchange. In 2014, Gregory Prisk, one of the world's leading experts on pulmonary physiology and altered gravity, published a review in the European Respiratory Journal explaining how microgravity alters lung function. He noted that gravity deforms the lung tissue itself through what he called the slinky effect. The weight of the lung compresses its lower portions and stretches its upper portions, creating different alveolar sizes and compliance at different heights. Remove gravity, and the lung becomes more uniformly expanded, but the system evolved to work with the non-uniformity. For a developing child on Mars, the lungs would develop in an environment with reduced gravitational deformation. The alveoli might develop more uniformly, with different size distributions than Earth lungs. The pulmonary vasculature might develop with less gravity-dependent variation. The ventilation perfusion matching might be calibrated differently. The respiratory muscles are another concern. Your diaphragm, the primary muscle of breathing, must push against the weight of the abdominal organs below it and expand the ribcage against the weight of the chest wall. On Mars, these weights are all 38% less. The diaphragm and intercostal muscles would develop to only Mars standard strength. On Earth, these weaker respiratory muscles might not be able to generate adequate ventilatory pressures, particularly during physical exertion. Consider also the development of the airway itself. The trachea, bronchi, and bronchioles develop their size based partly on the flow demands and partly on the physical forces acting on the chest. A child who cannot breathe as deeply because respiratory muscles are weaker might develop slightly smaller airways. Over a lifetime, this could predispose to respiratory problems, especially if gravity ever increased. Now, let me turn to something that is often overlooked in discussions about space physiology: the immune system. Because what happens to immune function in reduced gravity is both concerning and poorly understood. Research on the International Space Station has consistently shown that immune function is altered in space. T cells, which are critical for fighting infections, show reduced function. Natural killer cells, which destroy virus-infected cells and cancer cells, show altered activity. The reactivation of latent viruses, like the herpes simplex virus and the varicella zoster virus, is common in astronauts, suggesting impaired immune surveillance. Multiple factors contribute to this immune suppression in space: stress, radiation, altered sleep, nutritional changes, and the microgravity itself. But there is growing evidence that gravity directly affects immune cell behavior. Immune cells are sensitive to mechanical forces. Their activation, migration, and function are influenced by the physical forces acting on them. For a child developing on Mars, the immune system would be educated and shaped in an environment with reduced gravitational forces, chronic stress of an enclosed artificial habitat, limited microbial diversity, and elevated radiation. The implications for immune development are genuinely unknown. But we know from Earth-based research that the early environment profoundly shapes lifetime immune function. Children raised in overly sterile environments develop more allergies and autoimmune conditions. Children exposed to diverse microbes develop more robust immune systems. What happens to a child developing in the ultra-controlled, low-diversity, reduced gravity environment of a Mars habitat? We simply do not know, but there are strong reasons for concern. And then there is radiation. Mars has no global magnetic field and a very thin atmosphere. It offers minimal protection from galactic cosmic radiation and solar particle events. On Earth, our magnetic field and thick atmosphere shield us from the vast majority of cosmic radiation. On Mars, radiation levels at the surface are approximately 40 to 50 times higher than on Earth's surface. For adults, this increased radiation exposure raises cancer risk over time. For children, whose cells are dividing rapidly as they grow, the implications are far more serious. Rapidly dividing cells are more vulnerable to radiation damage because they have less time to repair DNA errors before the errors are replicated. A child growing on Mars would accumulate radiation damage to their DNA throughout their developmental period, during the time when their genome is being replicated billions of times to build their body. A 2022 systematic review by Guo and colleagues in PMC examined radiation-induced carcinogenesis in space conditions. They noted that radiation-induced DNA damage and subsequent mutations are the initial events of radiation-induced cancer. The concern for children is not just cancer risk, but also the potential for radiation-induced mutations in reproductive cells that could be passed to future generations, and developmental abnormalities caused by radiation damage during critical growth periods. This is not just about bone and muscle. Radiation could damage the DNA in bone marrow stem cells, potentially affecting blood cell production. It could damage neural stem cells in the developing brain. It could affect the germ cells that will produce the next generation. The combination of reduced gravity and elevated radiation creates a double assault on the developing body that has no precedent in human experience. Let me now talk about something that connects all of these systems together and reveals the true scale of the problem. It is the concept of developmental plasticity and critical windows. Throughout childhood and adolescence, the body goes through specific time periods called critical windows or sensitive periods, during which particular systems are developing rapidly and are highly responsive to environmental input. Once a critical window closes, the system that was developing during that window becomes much less responsive to change. The architecture is set. The calibration is locked in. Different systems have different critical windows. Bone mass is primarily built between birth and the mid-20s, with the most rapid accumulation during puberty. The vestibular system is functionally calibrated in the first few years of life. The visual system has a critical window that largely closes by age 8. The cardiovascular system's basic structure is established in the first years of life. Neural wiring patterns are most plastic in the first 5 years. The problem for Mars-born children is that all of these critical windows would occur in Mars gravity. Every system would be calibrated to 38% of Earth's gravitational signal during the time when its architecture is being established. And once those windows close, the calibration is permanent, or nearly so. This is fundamentally different from what happens to adult astronauts. Adults go to space with fully developed systems that were calibrated on Earth. They experience degradation, but their baseline was set in Earth gravity. When they return, their bodies remember the Earth calibration and can partially return to it. A Mars child would have no Earth calibration to return to. Mars is their baseline. Earth would be alien to them in the most literal biological sense. Let me tell you about the NASA twin study, because it provides a remarkable illustration of how even a single year in altered gravity can change the human body at the most fundamental level. In 2015, NASA astronaut Scott Kelly launched to the International Space Station for a mission of 340 days. His identical twin brother, Mark Kelly, remained on Earth. This created an unprecedented natural experiment. Two humans with identical genomes experiencing different gravitational environments. The study was massive in scope, involving research teams from 12 universities conducting investigations across multiple biological systems. The results, published in 2019 in the journal Science by Garrett Backelman and colleagues, were remarkable. Scott experienced significant changes in gene expression, with more than 1,000 genes showing altered activity during spaceflight. His telomeres, the protective caps on the ends of chromosomes, became longer in space, then shortened rapidly upon return. His gut microbiome shifted. His cognitive function showed measurable changes. Inflammatory markers were altered. Denamethylation patterns, which are epigenetic modifications that control which genes are active, were significantly different between the twins. Most changes reverted to baseline within 6 months of Scott's return to Earth. But 7% of his gene expression changes did not return to baseline. 7% of genes that changed their activity in space remained altered even after months back on Earth. Now I want you to sit with that number. 7%. After 1 year. In an adult whose biology was fully established before going to space, whose epigenetic patterns were already set, whose gene regulation was already stable. What happens when an entire developmental period, from birth through adolescence, occurs in reduced gravity? When the epigenetic patterns are being established, not just altered, in a non-Earth gravitational environment. When every gene regulatory decision is made in the context of Mars gravity. The scope of potential changes is orders of magnitude beyond what the twins study revealed. Epigenetics is the system by which cells decide which of their genes to activate and which to silence. It is what makes a bone cell different from a liver cell, even though they contain identical DNA. These epigenetic patterns are established during development, influenced by both genetic programs and environmental signals. Gravity is one of those environmental signals. Mechanical loading on cells influences their epigenetic state, determining which genes are expressed and at what level. A child developing on Mars would establish their entire epigenetic landscape in the context of Mars gravity. Their bone cells would have epigenetic patterns tuned for Mars-level loading. Their muscle cells would have gene expression profiles appropriate for Mars-level demands. Their cardiovascular cells would be programmed for Mars-level circulatory requirements. Every tissue, every organ, every system would carry the epigenetic signature of its developmental environment. And epigenetic patterns, once established during development, are remarkably stable. They can persist for life. They can even be passed to the next generation through a process called transgenerational epigenetic inheritance. This means that not only would Mars-born children be physically adapted to Mars, but their children and grandchildren might inherit some of these Mars-adapted epigenetic patterns, even if somehow conceived and developed on Earth. The effects could echo down through generations. Let me bring all of this together now with a thought experiment that I think will make the full scope of the problem viscerally real. Imagine 2 children born on the same day in 2050. One is born in a hospital in Toronto, Canada. The other is born in a medical facility in the first Mars colony. Let us call it Musk City. Both children have average genetics for a healthy human. Both receive excellent nutrition, excellent medical care, and loving families. The only difference is gravity, 9.81 meters per second squared versus 3.72 meters per second squared. By age 1, the Mars child is slightly different. their fontanelles may still be slightly more open, because the intracranial pressure dynamics are different. Their muscle tone is noticeably less, because they do not need to fight as hard against gravity to hold up their head or maintain sitting posture. They hit motor milestones like sitting and standing, but the effort involved is less, and the developmental stimulus to the brain from these achievements is different. By age 5, the differences are becoming more apparent. The Mars child is slightly taller than their Earth counterpart. Their limbs are slightly thinner. They are fully functional and active on Mars, running and playing, but an experienced pediatrician comparing them to Earth standards would notice reduced muscle bulk, particularly in the legs and core. A bone density scan would show values significantly below Earth norms for age. By age 10, the Mars child is noticeably taller and thinner. Their face may be slightly fuller due to the mild cephalad fluid shift. Their posture is different, with less pronounced spinal curves. Their movement patterns are graceful in Mars gravity, but would appear oddly light and underpowered in Earth gravity. They can jump remarkably high and far on Mars, because even their reduced musculature is sufficient to launch their body significant distances in low gravity. But the absolute force their muscles can generate is far below Earth child norms. By age 15, the Mars child is going through puberty. On Earth, puberty triggers the most rapid period of bone mineralization. Growth hormone surges drive rapid bone lengthening. Sex hormones drive bone densification. But all of this is occurring against the background of Mars gravity. The growth hormone surge produces lengthening, but the reduced gravitational signal means less densification. The Mars teenager grows tall, but does not mineralize adequately. Their peak bone mass, which should be rapidly accumulating during these years, is reaching only a fraction of Earth normal. By age 20, the Mars child is now a young adult. They are perhaps 10 to 15 centimeters taller than their Earth counterpart. They are thinner, with less muscle mass, particularly in the lower body and core. Their bone density is dramatically below Earth norms. Their heart is smaller and generates less pressure. Their blood vessels are thinner-walled. Their spinal curves are less pronounced. Their vestibular system is exquisitely calibrated for Mars, but would be overwhelmed by Earth. They are a healthy, functional, beautiful young adult, perfectly designed for their world. And they can never visit Earth. This is not a minor limitation. This is not like saying a person from Denver cannot live in Miami because they are not used to the humidity. This is a fundamental, structural, permanent incompatibility between their body and Earth's gravity. Their bones would fracture. Their heart would fail. Their balance would be impossible. Their spine would compress and herniate. Earth, the homeworld of their species, the planet where all their ancestors evolved, is as deadly to them as the vacuum of space. They are Martian. Not metaphorically, not culturally. Biologically, physically, irreversibly, this brings us to what some scientists are calling the two-species problem. Over multiple generations of Mars-born humans, if the colony persists and grows, the physical differences would accumulate. Not through traditional genetic evolution, which requires many thousands of years, but through developmental plasticity and possibly epigenetic inheritance, which can produce observable physical changes in just 1 or 2 generations. First-generation Mars-born humans would be different from Earth humans. Second generation, born to Mars-adapted parents, raised in Mars gravity, potentially inheriting some epigenetic adaptations, would be even more different. Third, fourth, fifth generation, with each generation never experiencing anything other than Mars gravity, the divergence would grow. At what point do we say these are still the same species? Biologically, species are often defined by the ability to interbreed and produce fertile offspring. If Mars humans and Earth humans cannot physically travel to each other's planets, if they cannot even survive in each other's gravity, does the question of whether they could theoretically interbreed even matter? They would be reproductively isolated by physics, if not by biology. This is not evolution in the traditional sense. Nobody is being selected for survival. Nobody is dying and leaving more or fewer offspring based on fitness. This is development shaping bodies to their environment in real time, every generation, with the results potentially being inherited by subsequent generations. It is a kind of rapid physical divergence driven by developmental plasticity rather than natural selection. Some researchers have suggested that within 5 to 10 generations, perhaps 200 to 300 years, Mars humans could be recognizably different from Earth humans. Taller, thinner, with larger heads, weaker musculature, different facial proportions, different postural geometry. They would still have human DNA. They would still be Homo sapiens genetically. But phenotypically, physically, they would be a different kind of human. Perhaps we would call them Homo sapiens Martianus. Or perhaps, eventually, just Martians. Scott Solomon, an evolutionary biologist at Rice University, published a book in February 2026 through MIT Press titled Becoming Martian: How Living in Space Will Change Our Bodies and Minds. In it, he explores how the human body and mind would adapt to life on Mars over generations. He compares the cultural and biological divergence of Mars colonists to the experience of immigrants who gradually lose their connection to their homeland over generations. By the 4th generation, he argues, Mars-born people would likely consider themselves fundamentally Martian, with Earth being as foreign to them culturally as it would be physically. But here is where the ethical dimension becomes impossible to ignore. Every child born on Mars would be born into this limitation without their consent. They did not choose to be born on a world that would permanently shape their body in ways that trapped them there. Their parents made that choice for them. And unlike almost any other parental decision, this one is irreversible and absolute. You can raise a child in any country on Earth, and they can later choose to live elsewhere. You can raise a child in any culture, and they can later choose to adopt different customs. But you cannot raise a child in Mars gravity and later give them the option of living on Earth. The physics does not permit it. A 2016 paper by Schuster and colleagues published in PMC, provocatively titled, Mars Ain't the Kind of Place to Raise Your Kid, explores the ethical implications of reproduction on Mars. The authors argue that bringing children into existence in an environment known to be harmful to their development raises profound ethical questions that we have never faced before as a species. On Earth, we have extensive regulations about exposing children to harmful environments. We do not permit children to work in toxic conditions. We do not permit children to be exposed to excessive radiation. Yet Mars colonization would, by definition, expose children to reduced gravity, elevated radiation, confined spaces, and limited medical resources from the moment of their conception. The question is not whether this is harmful. The science tells us clearly that it would alter development in profound ways. The question is whether those alterations constitute harm if the child is functional and healthy within their Mars environment. If a Mars child is happy, healthy, active, and well-adjusted on Mars, but can never visit Earth, is that harm? Or is it simply a different kind of normal? This is a question that science alone cannot answer. It touches on philosophy, ethics, autonomy, and what we believe we owe to future generations. But what science can tell us, and what this discussion makes clear, is that the decision to have children on Mars is not merely a personal choice like having children anywhere on Earth. It is a decision that permanently and irrevocably alters the physical capabilities and limitations of the resulting human being in ways that cannot be undone. Now let us talk about what we might do about this. Because the scientific community is not simply identifying this problem and walking away. There are proposed countermeasures, potential solutions, and areas where technology might help. But I want to be honest with you about the current state of these solutions. None of them are proven. Many are theoretical, and some raise ethical problems of their own. The first and most commonly discussed countermeasure is artificial gravity through centrifugation. The idea is simple in concept. Build a large rotating structure that simulates higher gravity through centrifugal force. You could potentially have children spend time each day in a centrifuge that simulates Earth gravity, providing the mechanical loading signals their bodies need for proper development. But the practical challenges are enormous. To avoid uncomfortable Coriolis effects, which cause dizziness and disorientation, a centrifuge needs to be large, at least 20 meters in radius for comfortable human habitation. That is a massive structure to build on Mars. And then there is the question of how much time children would need to spend in the centrifuge. Bones respond to continuous loading, not just periodic loading. The 24-hour gravitational signal that drives development on Earth cannot be replicated by a few hours in a centrifuge. Studies on Earth using vibration platforms and resistive loading in children with mobility impairments show that periodic loading can improve bone density, but it does not fully replicate the effects of continuous gravitational loading. The bone develops differently when the loading signal is intermittent versus constant. Think of it like this. Hearing music for an hour a day is not the same developmental stimulus as being immersed in a language-rich environment 24 hours a day. The periodic signal provides some benefit, but cannot substitute for constant environmental input. Would children need to sleep in centrifuges? Possibly. Would they need to spend 8, 12, 16 hours a day in higher gravity? Perhaps. At what point does the centrifuge become the dominant feature of the child's life? At what point are we essentially keeping children in a medical device for their entire childhood to compensate for a problem that we created by choosing to raise them on Mars? The second proposed countermeasure is pharmaceutical intervention. There are drugs that can influence bone metabolism. Bisphosphonates, for example, are used on Earth to treat osteoporosis by reducing bone breakdown. Anti-rankle antibodies like denosumab block the signals that cause bone resorption. Growth hormone supplementation could potentially drive more bone formation. Myostatin inhibitors, currently in experimental development, can prevent muscle loss. But using these drugs in children raises profound concerns. Children's bodies are growing and developing. The systems these drugs target, bone remodeling, muscle protein synthesis, growth hormone pathways, are operating at full speed during development, building the adult body. Interfering with these systems pharmaceutically in a developing child is vastly more risky than treating an adult with established physiology. The long-term effects of continuous bisphosphonate use in children are not well understood. Growth hormone supplementation in children without growth hormone deficiency can have serious side effects, and myostatin inhibitors have never been tested for chronic use in developing organisms. Furthermore, even if we could safely boost bone density and muscle mass pharmacologically, we would not be addressing the architectural and geometric aspects of skeletal development. You can make a bone denser by adding mineral, but you cannot change its shape, its cross-sectional geometry, its trabecular architecture, or its cortical distribution with drugs alone. The bone would still be structurally adapted to Mars loading patterns, even if artificially mineralized to Earth-like density. The third proposed countermeasure, and perhaps the most philosophically troubling, is genetic engineering. With technologies like CRISPR, we could theoretically modify the genes of children before or soon after birth to produce denser bones, stronger muscles, larger hearts, and other adaptations that might compensate for reduced gravity. We could potentially modify the Wnt signaling pathway to increase bone formation sensitivity, so that even the reduced mechanical signal from Mars gravity would produce near normal bone density. We could alter myostatin genes to make muscles resistant to atrophy. We could enhance cardiac muscle gene expression to produce a stronger heart even without Earth-level circulatory demand. But think about what we are proposing here. We are talking about genetically engineering children specifically designed for an alien environment. These would not be normal human children with supplementary treatments. These would be biologically modified organisms, designed from their DNA up to function on Mars. We are talking about creating humans specifically adapted for another planet. About deliberately choosing to deviate from the human genome as it has existed for hundreds of thousands of years. About making permanent, heritable changes to the human germline that would be passed to all future generations of Mars humans. Is this solving the problem, or is this creating a new species by design rather than by accident? The ethical implications are staggering, and unlike most genetic modifications we discuss today, which aim to cure diseases or fix deficiencies, this would be modifying healthy human genes to adapt to an artificial situation that we chose to create. We would be re-engineering the human body to fit a world rather than finding a world that fits the human body. The fourth proposed countermeasure is mechanical loading through wearable technology. Loading suits, weighted clothing, exoskeletons that constantly compress the body to simulate higher gravity. The idea is that you could strap additional mass to a child's body or use spring-loaded suits that resist all movement, creating resistance that mimics gravitational loading. This is perhaps the most practical of the proposed solutions, and research on Earth with loading suits shows some benefit for bone density in elderly populations and mobility-impaired individuals. But the magnitude of loading required is enormous. To simulate Earth gravity on Mars, a child would need to carry an additional 162% of their body weight in loading. For a 30-kilogram child, that means an additional 49 kilograms of resistance. Constantly, 24 hours a day. Through running, playing, sleeping, everything, can you make a child wear a nearly 50-kilogram loading suit for their entire childhood? Would this not create its own developmental problems? Joint stress, skin breakdown, restriction of movement, psychological distress? And would intermittent loading during the day, with the suit removed for sleeping and bathing, be sufficient? The data suggests it would not fully replicate continuous gravitational loading for the same reasons that periodic centrifuge use may be insufficient. The honest, scientifically rigorous answer to the question of whether we can prevent Mars gravity from deforming children's bodies is, we do not know. We have theoretical proposals. We have no proven solutions. We have never tested any of these countermeasures on developing organisms in partial gravity. We have never even tested them on developing organisms in full microgravity, because no mammal has ever been raised from birth to adulthood in space. We are discussing sending humans to Mars to create permanent settlements without having answered the most basic biological questions about whether human children can develop normally there. We are planning families on another planet without understanding whether those families can produce healthy children. This is not a failure of ambition. It is a failure of priorities. We are investing billions in rocket technology to get to Mars, while investing relatively little in understanding whether human biology can survive on Mars across generations. Let me now address something that I think is often overlooked in the excitement about Mars colonization. People often point out that Mars has 38% gravity, not zero gravity, and assume this means the problems will be only proportionally bad. 38% gravity should be 62% of the problem of zero gravity, right? That is intuitive, but biology does not work linearly. Remember the Swanenberg study from 2023. They found that Mars gravity induced similar changes in spinal motor control as microgravity. Not 38% of the changes, similar changes. This suggests there may be a threshold effect. Below a certain gravity level, certain developmental systems may effectively receive the same signal as zero gravity. The difference between 0.38 g and 0 g may be much less biologically significant than the difference between 1 g and 0.38 g. We see threshold effects throughout biology. A muscle that receives some stimulus is not necessarily proportionally better off than a muscle that receives no stimulus if the stimulus is below the activation threshold for the relevant physiological processes. A bone that receives 38% of the normal loading signal does not necessarily maintain 38% of normal density. If 38% is below the threshold for maintaining proper trabecular architecture, the result might be similar to zero loading. We genuinely do not know where these thresholds are for human development because we have never tested them. No human child has ever been raised in anything other than Earth's 1G. No one has been raised in 0.5G, or 0.7G, or 0.3G. We have exactly one data point. 1G works for normal human development. Everything else is extrapolation and inference. Some scientists have proposed that we need to determine the minimum gravity threshold for normal human development before we commit to permanent Mars colonization. If that threshold is, say, 0.5 g, then Mars at 0.38 g is below it, and permanent colonization without artificial gravity would be biologically untenable. If the threshold is 0.3 g, Mars might be just barely adequate. But we do not know. And finding out is incredibly difficult because simulating partial gravity on Earth for the duration of an entire developmental period is nearly impossible with current technology. There is one more aspect of this problem that I want to discuss, and it touches on the deepest implications for humanity's future. It is what I call the first-generation trap. The first adults who go to Mars will be volunteers. They will be informed, consenting adults who understand the risks to their health. They will experience degradation, bone loss, muscle loss, cardiovascular changes, but they were built for Earth and chose to accept the consequences of leaving it. Their bodies remember what they should be, even as they change. But the first children born on Mars had no choice. They did not volunteer. They were created in that environment by parents who may or may not have fully understood the consequences. And those children, once born, cannot be unborn. Their developmental trajectory, determined by Mars gravity from conception onward, cannot be undone. They are the first generation of a new kind of human, created without consent, trapped on a world not by walls or laws, but by the very structure of their own bodies. And here is the trap. You cannot know whether Mars-born children will be healthy until they exist. You cannot run the experiment without creating the experimental subjects. And those subjects are human children. This is not like testing a new drug where you can start with low doses and escalate carefully. The experiment is all or nothing. Either you allow a child to be conceived and develop in Mars gravity, or you do not. There is no half measure. There is no way to partially expose a developing fetus and child to Mars gravity to see what happens. Some have suggested using animal models first. Raise mice, or larger mammals, from conception to adulthood in Mars simulated gravity to see what happens. This is scientifically sound, but practically challenging. Simulating Mars gravity on Earth for the duration of an animal's developmental period is technically possible using centrifuges running at specific speeds, or using partial weight-bearing systems. But doing so for months or years with adequate controls is expensive and complex. Some limited animal studies in actual space have been conducted. The pregnant rats on the space shuttle in 2008 showed abnormal vestibular development in their offspring. Mouse embryo studies published in 2023 showed that early embryonic development can occur in microgravity. But the embryos were not developed to term. We have fragments of data, pieces of the puzzle, but not the complete picture. What we need, and what the scientific community increasingly recognizes we need, is a dedicated program of partial gravity developmental biology research. We need to raise animals from conception to full maturity in Mars-equivalent gravity. We need to assess their skeletons, their hearts, their brains, their vestibular systems, their muscles. We need to test whether they can survive a transition to Earth gravity. We need to do this with multiple species, over multiple generations, before we even consider allowing human reproduction on Mars. This research could be done on the International Space Station, or its successors using centrifuges to create Mars-equivalent gravity within a space habitat. It could be done on the planned Lunar Gateway Station. It could be done on the Moon, which at 0.17 G provides an even more extreme partial gravity environment for testing. But it needs to be done. And it needs to be done before the first human child is conceived on Mars, not after.